for joining us for the first General Club for 2024, which is the fifth one in our GSU monthly webinar series. And the talk will be for about 40 minutes or so. And at the end, we're going to have 20 minutes Q&A as well as comment time. So please do type your questions and comments in the boxes below. There's a chat box and a Q&A box. So please choose either of those and type your questions and comments as the presentation is unfolding. We are excited to kick off the new year with a malaria parasite focused General Club with Dr. Zach Popkin-Hall, as you can see on the screen right now. And the title of the talk is Characterization of the Malaria Species Landscape in Mainland Tanzania. The presentation is based on a recently published first author paper in the Journal of Infectious Diseases by Dr. Popkin-Hall et al. Quick bio to introduce our speaker today. Dr. Zach Popkin-Hall is a postdoctoral research associate in the infectious disease epidemiology as well as ecology lab at the University of North Carolina. His primary research focus is non-falciparum malaria genomics, and he has research collaborations with colleagues in Tanzania, Cameroon, as well as Peru. Without further ado, thank you so much, Dr. Zach, for honoring our general invitation. Please go ahead and give a presentation.
All right, well, thank you so much for inviting me, Pablo, and everyone at Sanger. It's a pleasure to be here and an honor to kick off the first Journal Club of 2024. So like you heard, I'm going to be talking today about our results from mainland Tanzania, characterizing the malaria species landscape there in 2021. This is work that was done in collaboration with us at IDEAL, at UNC, and the School of Medicine, as well as with our collaborators at NIMRI, the National Institute for Medical Research in Dar es Salaam, and at Brown University, also in the US. So I'm just going to first give an overview of what I'm going to talk about today in this presentation. So first, I'm just going to explain a little bit about the project that generated this research, which is known as MSMT. We'll spend the bulk of the time talking through the paper that landed me this invitation, which is this 2021 survey of patients at health facilities in mainland Tanzania, which is now out in Journal of Infectious Diseases, as you heard. I'll also talk a little bit about a complementary study that took place at the same time, looking at community surveys, and that manuscript is currently in review at Parasites and Vectors. I'll talk very briefly about data that we have from 2022, because this will be a longitudinal study, as well as one that looks at the data from 2021. And then I'll shift gears and talk genomics, which is really the direction that we're moving towards with this data. So I'll present some preliminary data from sequencing that we've done at Plasmodium Malaria, as well as preview a little bit of work that we're going to do on Plasmodium ovale. And then we should have plenty of time for questions at the end. So I want to first just give a sense of where this research is coming from. So this is coming from a Bill and Melinda Gates Foundation funded project called the Molecular Surveillance of Malaria in Tanzania. This is a collaboration between our PIs on the far left of this photo, Deus Ishingoma at NIMRI, Jeff Bailey, who's standing next to me at Brown, and then my advisor, whose office I'm in right now, John Giuliano, who's sort of looming in the back on the right side of this photo. And then we have many additional collaborators at CDC and various other places. And the goal of this project first is to characterize and monitor malaria parasite populations in mainland Tanzania, looking at 3 foci within that. So characterizing anti-malarial resistance, characterizing HRP2 and 3 gene deletions to look at the prevalence of diagnostic evasion, and then the focus of my talk and my work with this project to look at the species distributions in mainland Tanzania. More recently, this project has also added another goal, which is or 2 other goals, to monitor the vector populations in Tanzania, as well as to monitor human variants of interest, so things like G6PD. that are related to malaria resistance and clearance. And in addition to that, we have these sort of larger goals, which are to build capacity and local molecular and genomic capabilities within mainland Tanzania to support improved surveillance and responsiveness, diagnosis, and treatment. So in addition to the scientific goals, this project is really focused on building a state-of-the-art and self-sustaining lab in Dar es Salaam to be able to do this work eventually without me and others like me having to contribute to it. Although, of course, I'll be happy to if they ask me to. And all of this is in service of the larger goal of accelerating progress toward reduced malaria morbidity and mortality in line with the stated goal of malaria elimination. So I'm also going to spend a little bit of time characterizing how malaria looks in mainland Tanzania. So this is data from 2021. So in 2021, Tanzania had the fourth highest malaria mortality rate worldwide, but it has very heterogeneous transmission. So if you look at the map that I have on the right, this is data that I have from the National Malaria Control Program based in Dodoma. And it's showing the malaria prevalence. This is just everything from RDTs and microscopy for each region of Tanzania. And you'll notice that in some regions, we have quite a lot of malaria transmission where the dark blue is. And then in other areas, like in the interior, there's very little transmission at all. So the picture in Tanzania is not at all uniform. And I should also make a disclaimer here that I'm talking exclusively about mainland Tanzania. And the situation in Zanzibar is very different. So in Zanzibar, it's actually already a pre-elimination setting, and Zanzibar maintains its own malaria control program, independent of what the NMCP in Dodoma does in the mainland. So in the mainland Tanzania, most of the malaria is Plasmodium falciparum, as is true in most of Africa. And in terms of control and treatment at the moment, Tanzania is estimated to have greater than 50% coverage with long-lasting insecticide-treated nets or indoor residual spraying for mosquito control. And then in terms of medication, the first-line treatment is artemether-lumefantrine, and then there is some IPTP with sulfadoxine and pyrimethamine. And then the last thing I wanted to show here, just to characterize the malaria situation in Tanzania, is that these graphs from the World Malaria Report showing sort of the trend of malaria cases and deaths in Tanzania. So if you look at the blue line, which is cases, in 2000, Tanzania started with a very high malaria case burden of 12 million cases a year. That has reduced quite a lot since 2000, but you'll notice, and I'm sure this is not new for most of you, that those cases have gone up again a bit. And we've seen a bit of a plateau, and then there was an increase associated with lockdown in 2020 and the interruption of malaria control measures. And then if we look at the right, we see malaria deaths. So again, a very high mortality rate in 2000 that again has gone down significantly, but has risen again a little bit. So obviously we want to get those cases and deaths back down to achieve our goal of malaria elimination. So that's the situation in terms of how much malaria there is in Tanzania. Now I want to talk a little bit about the species that I'll be talking about today. So I think everyone who works in malaria is very familiar with Plasmodium falciparum, and many of us are less familiar with the other species that can cause malaria. So in Africa, there are 4 others that are present. The first of which is Plasmodium malariae, which is sort of, I would say, the least understood, can cause these long-term persistent infections. Then we also have the 2 Plasmodium ovale species, Curtici and Willikeri, which used to be one species, have since been split. And both of those cause relapsing malaria due to the presence of hypnozoites in the liver. And in addition, there's also a degree of Plasmodium vivax in Africa, which again causes relapse because of the hypnozoites. All of these non-falciparum species tend to be less severe, and they tend to cause lower parasitemias than falciparum, but all of them can still cause serious complications and mortality. But that said, they tend to co-occur with Plasmodium falciparum, especially in the African context. And the big worry and sort of the impetus of this project is that those non-falciparum species may become more prevalent as falciparum is eliminated, which is something that we have seen in other parts of the world. Um, so as additional context, I want to talk a little bit about another study that was done in mainland Tanzania looking at the malaria species landscape. So this is work that was done by a PhD student in our lab, or the manuscript at least was, Rachel Sender. And this is analysis of the 2017 National School Children Survey, which surveyed children in schools in Tanzania between ages 5 and 16. And what was really striking about this study is that it showed actually that the overall prevalence of Plasmodium ovale was similar to that of Plasmodium falciparum. So if you look at this diagram that I have on the right here, you'll see there actually are more ovale cases detected than falciparum. What's also interesting is that the majority of those, 64%, were single species infections. So just ovale, not co-infected with falciparum, although we do still see a large share of co-infections. In addition, this study did find a smaller degree of Plasmodium malariae infection, most of which were co-infections, and then a very small number of Plasmodium vivax cases, so a very low prevalence of vivax. So that's what we knew going into this study, but this was just in school children. No one had done this looking at like the broader population. So in terms of our methods, these samples were collected, like I said, in 2021 in health facility surveys. So all of these samples came from symptomatic patients with malaria-like illness that were seeking care at health facilities in 10 different regions, which are labeled in the map on the bottom right. We collected a total of 12,845 dried blood spots from patients at those health facilities. And then we drew a random subset of 3,284 of those, which are then what we analyzed to determine the positivity rate of all of these different species of Plasmodium. And I'm not expecting anyone to read this in great depth. If you would like to, it is in the manuscript. But this is just showing you the numbers of the samples that we collected in each region. It also shows the age distribution within each region, the gender distribution, and then the proportion of those samples that were RDT positive, meaning that they were likely to have Plasmodium falciparum. And the other thing I do want to point out about this table is that these regions come from a variety of different transmission strata. So we had 3 regions that were considered to be very low transmission, 3 that were considered to be low, 2 that were considered to be moderate, and 2 that were considered to be high. And those transmission strata were determined from 2020 data, which I think was actually collected in 2019. So all of that, which is to say that those strata may not necessarily correspond to the situation in 2021. And also that those strata are determined by Plasmodium falciparum transmission intensity, so not necessarily expected to match what we see in other species. So I want to tell you a little bit more about how we did this analysis. So this was really facilitated by using these Opentrons liquid handling robots that we have installed at NIMRI and Dar es Salaam, which are able to both replate the samples that we chose to analyze, as well as to set up our plates for qPCR for us by adding the master mix. So that makes things run a lot faster, a lot more accurately. And then on the right, I'm just showing you a sample of what our qPCR curves look like. So to do this analysis, we used a semi-quantitative 18S qPCR using serial plasmid dilutions. So we're running a separate qPCR for each species. So I'll now show you some of our results. So the first thing I'll show you is our positivity by region for each species. So on the left, again, here is our locator map showing which regions these are, since not everyone will be very familiar with Tanzanian geography. And then on the right, I have the maps for each species. So on the top left, figure A, we have Plasmodium falciparum. And what will jump out to you right away there is that these positivity rates go up to about 80%. So Plasmodium falciparum accounts for a large proportion of the patients who had malaria in this cohort. And you will again see some heterogeneity in terms of where falciparum is most abundant. So we see the heaviest or the highest positivity rates for PF here in Tabora region as well as in Mtwara region. And then to the right of that, the top right figure B in purple, we're looking at Plasmodium malariae. So the positivity rates there only go up to a little over 4%, quite a bit lower than falciparum. But we do see, again, that regional heterogeneity. So we see the highest positivity rate for PM in Mara region along Lake Victoria. And then if we look at the bottom left in the green, we're looking at Plasmodium ovale. Here I'm looking at both Curtici and Willikeri. We did not speciate to determine which ones this was. But the ovale positivity rates are quite a bit higher than for Plasmodium malariae, although still lower than falciparum. So PO positivity goes up to about 15%. And again, we're seeing that highest positivity in Mara region with, again, some heterogeneity elsewhere. And this will be clearer on the next slide, but we actually see that the ovale positivity rates surpass 5% in 7 of our 10 study regions, which is suggesting that really there needs to be some targeted surveillance specifically looking for ovale, since it accounts for such a large proportion of infections here. And then finally, in the bottom right, in the sort of burnt orange color, I'm showing Plasmodium vivax. And this map may be a little misleading. It kind of looks like there's vivax in all of these regions, but in fact, what that's showing is that in every region but one, there's 0 vivax detected. So we only detected 3 total cases of PV, and all of them were in Kilimanjaro, which is considered to be a very low transmission region. So an interesting situation there in Kilimanjaro. So I'm also going to show you this in tabular format because I think it makes a couple of things clearer. So for instance, I've highlighted the regions that are particularly high in positivity rate for PM and PO. And you'll see, so in Mara, that's really the only region where PM gets anywhere near that 5% mark. But then for PO, like I was saying, 7 out of our 10 regions are at 5% or above. So that's less PO than was found by the previous study of school children, but it's more than I think was known to exist in Tanzania previously. But all of this in context, there is quite a bit more PF than there is of either PM or PO, and very little vivax. And then we also wanted to look at what the composition was of those infections. So on the top of this table, I'm showing our malaria results. And so I have counts as well as proportions and infection types. So the majority of our PM infections are co-infected with Plasmodium falciparum. That accounts for a little more than half of them. Although we do still have about a quarter of those infections, which are single species infections of PM. And then for PO, which is right below there, a little more than 3/4 of them are co-infected with PF. And a smaller percentage, about 15%, are single species infections of just PO. And I don't want to spend a lot of time on this, but I will also mention that we do have 1 co-infection of just PM and PO, and then 10 triple infections where these patients had all 3 at the same time. And then finally, on this slide, I want to point at the bottom to our Vivax results, which show that of all 3 of our Vivax positive samples were co-infected with falciparum. So there's no single species infections of Vivax detected in this study. So next, we wanted to do some statistical analysis looking at variability of our positivity rates by transmission strata. So like I mentioned, these transmission strata were determined in 2020, so not based on our 2021 data, so they wouldn't necessarily line up. On the left, with letter A, we're looking at PF, and just the positivity rates for the different transmission strata. So you'll notice actually that our moderate transmission strata has the highest positivity, and that is significant, and we do see significant differences between all of our strata. And probably that is explained by the transmission levels in 2021 being different from what they were when these strata were determined. In the middle, we're showing PM, and we'll see that that's even more dramatically the case, that really we see a lot of transmission in the moderate transmission strata, although the difference between moderate and high for PM is not significant. But then again, if we look at PO Valley on the right, we do see that that difference is again significant. So in all three species, the moderate strata seems to have the highest transmission, and there are significant differences between moderate and the low and very low. And I should also say all of these are symptomatic patients, so it's not terribly surprising that it largely tracks with the transmission intensity. In addition to transmission strata, we wanted to look at variability by age group. So this is to sort of put our results in context from that previous study that had looked at school children. So I've divided the samples into 3 different age groups: young children under 5 years old, school children based on the ages from that previous study, so children between 5 and 16 years old, and then adults, which we here are characterizing as anyone 16 years or older. So on the left, I'm looking at, again, at Plasmodium falciparum, and we see there that school children have significantly higher rates of Plasmodium falciparum than either young children or adults. Not too surprising, given that there is a large body of literature saying that school children tend to be a reservoir for malaria. They tend to have a lot of it. But we do see that trend also in Plasmodium malariae in the middle there, and in addition in Plasmodium ovale on the right. And so that may partially explain what's going on in that other study that looked exclusively at school children, since we do see that school children seem to have significantly higher rates of Plasmodium ovale here than the adults or the younger children. So to put this all in context, I'll add a bit of discussion points. So first and foremost, I want to say, what we're getting from this data, which is we have this comprehensive picture of the malaria species landscape among symptomatic patients, at least in mainland Tanzania. And what we see is that Plasmodium falciparum is still the most prevalent malaria species in Tanzania by quite a lot, which suggests that the NMCP is right to continue to focus primarily on PF. But we do see that Plasmodium ovale surpasses a 5% positivity rate in 7 out of our 10 regions that we studied, which indicates that we do need some proactive monitoring and potential implementation of new control measures to focus on ovale, given that it causes relapse and can sort of persist even when there's not active mosquito-borne transmission. Malaria is less common than either PF or PO, but still warrants surveillance, given that we do see it at those low levels kind of all over the place. And PV, however, is still very rare, and we only found it in those 3 samples in Kilimanjaro region. So for now, at least, it seems like in Tanzania, PV has a very minor clinical impact and is not a major consideration for control and elimination activities. We did see that positivity rate varies significantly by transmission strata, which did not correspond to that earlier transmission intensity. But like I mentioned, that's probably mostly a factor of those strata being picked before this data was collected. And as I was saying, the school children are significantly more likely to test positive for all 3 species of malaria. Most of our non-falciparum infections were mixed with PF, which is different than what was found in that other study. But again, these are symptomatic infections, and it includes all age groups, not just school children. In addition, so there's currently 2 active case-based surveillance pilots for elimination in Kilimanjaro and Manyara regions, which are 2 of the low transmission regions we included in our study. So to put our results in the context of that, non-falciparum species seem to be quite rare in Kilimanjaro. So there, at least, I think that they're unlikely to present a major barrier to doing the control and eventual elimination. But in Manyara, we did see that PO levels are greater than 5%. So there's probably a need in Manyara to incorporate radical cure with primaquine or tafenoquine to get rid of those hypnozoites, as well as to do active surveillance for relapse cases of malaria there in order to effectively control malaria in that region. There are some limitations to this study. So something we'll address in just a minute here is that this study did not include asymptomatic patients. So these are not true prevalences, they're just positivity rates. So it's not a full characterization of the species landscape in those regions. We also have an additional limitation, which is that RDT positive patients were likely over-enrolled. So even though other people may have come to the clinic with febrile illness and tested negative, probably fewer of them were included in the study for a variety of reasons. And in addition to that, malaria-like symptoms, as I'm sure many of us know, don't necessarily reflect malaria infection. So because we're also looking at RDT negative individuals, patients who tested positive for non-falciparum infections, that may not have been why they had febrile illness. They may have been infected with respiratory illnesses or arboviruses, things of that sort, and we did not sample or test for any of those. So we don't necessarily know why these people are sick, even though they are infected with malaria. And an additional limitation is that this study was not fully nationally representative. And as I mentioned, it does not include Zanzibar, which is the pre-elimination region in Tanzania. In conclusion for this part of the presentation, for this study, we've established this important baseline that will enable us to characterize temporal trends with the incorporation of data from 2022 and 2023, which we are actively analyzing now. So now I'm going to move into things that are not in that published paper, so you may not have seen any of this before. So in addition to the symptomatic patients at health facilities, we also did community surveys at households in 3 regions of asymptomatic cases. So we took a subsample of 1,428 samples that were drawn randomly from about 6,000 dried blood spots collected in 2021. So like I said, in 3 regions, Kigoma, Tonga, and Ruvuma, we did these household surveys of asymptomatic individuals. And then in addition, we're also incorporating data from a separate study of children under 5 in Geita region. And just from looking at the map of the transmission on the right side of this slide, you'll see that these are all higher transmission areas. And here, I'm just showing you a table to give you a sense of the sample numbers, but also to show you those transmission strata. So Tonga was considered to be a moderate transmission region. Geita, Kigoma, and Ruvuma were all considered to be high transmission. And this is also just showing you roughly how many samples came from each of those regions. So here I'll show you the results in terms of our regional infection prevalences, as well as our infection compositions. So we did not detect any Plasmodium vivax in this asymptomatic survey, so there's no vivax map. So instead, what I've done is I have the region locator in the top left, and then our 3 species that we did detect. So again, you'll notice that PF is the most abundant species, with prevalences going above 36%. In Tonga and Geita, it's especially high, not so high in Kigoma and Ruvuma, but still fairly high, above 20%, especially considering these are all asymptomatic individuals. In the bottom left, we have PM. So this is fairly lower than we see for PF, for example, but we do still see that it ranges between 1% and 3% in each of those regions. So there is definitely a substantial amount of Plasmodium malariae circulation. And then on the bottom right, we're again looking at Plasmodium ovale. And with the asymptomatic patients, we see that the prevalence for PO is pretty similar to what it is for PM, a little bit higher. So surpassing 3.6% in Kigoma and ranging a little bit between 2.5 and 3 in our other regions. And then again, we also wanted to look at the infection composition to see how frequently these infections were co-infected with falciparum. So on the top, again, we're looking at PM. And what's interesting here is that we actually see a different trend than we did in the symptomatic patients. So we have a slight majority of PM infections, or not a majority, a plurality, that are single species infections, so just PM, not infected with anything else, although there are a very similar number of PM infections that were co-infected with PF. And then if we look at PO, we see a more similar trend to what we saw with the symptomatic patients, where most of them are co-infected with PF, but we do see a larger proportion of these asymptomatics that are single species infected with just PO. And we do again have a small number of triple infections with all three species. And as I mentioned, no Vivax was detected in this survey. We again looked at variability by age group. And this again is sort of to allow us to more directly compare to that previous study that had shown such high rates of ovale in school children. So with falciparum, which we'll look at first, on the left, you'll see that again, we see that school children have the highest prevalence. There is a significant difference between the school children and the adults. The difference between the school children and the young children is not significant, but we do see the same trend that we saw before, where the school-age children tend to have the most, and then children generally have more than adults do, which again, for falciparum, that is well established in the literature. For PM, though, we see something quite different from what we saw previously in our symptomatic patients, which is actually that we have a significantly higher likelihood of being infected with PM in adults than we do in young children. And that trend is also present between adults and school children, although not significant, which is likely due to these small sample sizes. But what we think is likely driving the adults to have this higher prevalence in PM is the presence of those long-term persistent infections that I mentioned at the beginning of the talk. Because these adults are older, they've probably had more opportunities to get infected with PM, and so they may have been infected years ago and still have PM circulating in their blood. And then finally, we show PO on the right. There's no significance there, but we do see the same trend that we see in PF, and also that we saw in PO in the symptomatic patients, and that is in keeping with that previous study of school children showing that they seem to be the most likely to be infected with PO. So discussion for this analysis, this really serves as a complement to the health facility data because it helps us to look at the asymptomatic patients as well as symptomatics. We do again see that PF is the most prevalent species, but we also see that even in asymptomatic patients, we found PM and PO in all of our regions, with each of them surpassing 3% prevalence in at least one of those regions. PO, as I mentioned, was mostly co-infected with PF, but PM, I say majority here, but again, that's actually a plurality, are single species infections. And children are more likely to have PF and PO, but not PM, which again, we think probably reflects that persistent infection. But I do want to add some caveats, which is that these are pretty small sample sizes. And in addition, the subsample we actually determined is not representative of the overall age structure or gender distribution. So there may be some inconsistencies there. And in addition to that, our samples that came from Gaeta, all of those are from young children. So the Gaeta samples are just sampling a totally different study population than we got from the other three regions. But in conclusion, this study serves, like I said, as an important complement to the health facility data in helping us build this really comprehensive baseline picture of the malaria species landscape in the mainland of Tanzania. So I'll give you just a really quick preview of our data that we have from 2022. The 2023 analysis is ongoing. So in 2022, we haven't done any of these detailed analyses, but we did find 67 samples that were infected with PM in 12 out of 13 regions. The highest positivity rate was in one of our high positivity rates from 2021, Kagera, at 4.1%. Then we had 141 PO positive samples from all 13 regions, with the highest positivity rate again being in Mara, where it was in 2021, although it is quite a bit lower at 6.7% instead of 15%. But that's still above 5% and still well worth actively surveilling. And in addition to that, I want to point out that we pulled 90 of these samples to do our genomic work on PM and PO, which those analyses you'll hear more about in just a minute. And there's going to be a lot more analysis done of this data along with the 2023 data. So we'll be able to start characterizing temporal trends, which I think is really the most important thing that we can get out of this study. And in 2023, I also want to note that data were collected from every single region of mainland Tanzania. So the 2023 data will give us really a full picture of what's going on with malaria all over mainland Tanzania. But I do not have data to present from that yet. So now I'm going to shift gears entirely and talk about some of our genomic work. This is in earlier stages, so it's not as complete of a picture, but it is still some interesting stuff to talk about. And since I have the chance, I figured I would. So like I mentioned, we pulled some of those low CT, high parasitemia samples for sequencing of PM and PO. So this is data that I'm presenting on some of those 2021 PM samples. So on the left here, I'm showing a map of just the regions within Tanzania where we drew samples from, just showing that we have a pretty good geographic spread throughout mainland Tanzania. So that included 50 samples that came from this study, and then some that also came from another study in Tanzania led by Jessica Lin, who's here at UNC, and Billy Nagasala, who's at MUHAS in Tanzania. They were kind enough to let us look at their samples as well. And then we also are including samples not from Tanzania. So we have 16 samples from the DRC, 5 from Nigeria, and 5 from Cameroon that are also included in this study. Those have not been as fully analyzed as the Tanzanian samples, full disclosure, but I will mention them here and there. So to process these samples, we did a custom hybrid capture library prep to enrich for PM sequence as opposed to PF or human, since these are blood samples. And then we sequenced them on an Illumina NovaSeq platform here at UNC. I used a bioinformatic pipeline that filtered out PF and human reads using this tool called bb-split before I did variant calling and analysis. And the first thing I want to show you is just how well we were able to enrich for PM. So what this is showing in this figure is the proportion of reads mapping to the PM genome as opposed to PF or human. So the bright orange bars are PM, the light orange are PF, and then the yellow is human. And so this is each of those 50 samples from Tanzania. Each bar is a separate sample. And then the y-axis is the percent of reads that map to that particular genome. So you'll see there is definitely some variability. Some of our really well-performing samples, we had well above 75% of the reads mapping to the PM genome. We do have some less well-performing samples where it's closer to 1% mapping to PM. But to put that in context, if we had tried to do direct sequencing just from the blood sample, we would have, almost everything would have looked like this one at the far right, where it would have been almost entirely human due to the high amount of human DNA in the blood sample. So what that tells us is that even though there's some variability, this hybrid capture method works well to generate PM sequences. And 29 of these 50 isolates produced over 1 million mapped reads that map to PM. And unsurprisingly, the performance was highly negatively correlated with CT, meaning that our high parasitemia samples performed well, our low parasitemia samples did not perform as well. Not too shocking, I don't think. We also wanted to just characterize how well we did at genome coverage using this sequencing method. So what I'm showing here is box plots of sequencing coverage across the genome, including the 14 assembled chromosomes, as well as the apicoplast, excluding unassembled extrachromosomal contigs. And I don't believe there's an assembly either for the mitochondrion for PM yet. So just this is including those 14 chromosomes in the apicoplast. Coverage is generally good. So you'll see that our medians tend to be around 50x or above, but it is variable. 45 of these 50 samples from Tanzania had greater than or equal to 10x coverage across all of these chromosomes in the apicoplast. and then 18 had greater than or equal to 50x coverage across all of the chromosomes in apicoplast. So that should give us a very good sample to really do some variant calling and get a good picture of what's going on with PM genomics in Tanzania. So now I'm going to show some of our more interesting results now that we've established that the method works. So I'm showing principal component analysis, and I'm showing two different versions of this. So on the left, we're just looking at Tanzania, and what I've done is I've color-coded each sample by region. So what you'll notice right off the bat, if you're used to looking at PCA plots, is that there's basically no structure there. And in addition to that, our principal components explain very little variation. So the first two combined explain a little over 6% of variation. So then I was curious to see if we pulled in our samples from other countries that are not nearby, so Cameroon, DRC, and Nigeria, would we get any structural resolution? And so that's on the right where we have color-coded by country, and you'll notice again, no geographic structure. So there really seems to be very little geographic structure, at least in Africa, among PM isolates. And to put this in context, I want to compare it to work that we've done on Plasmodium ovale. So this was done by an MD PhD student in our lab, Kelly Carey Ewand. So on the left, you'll see my PM PCA. On the right, you'll see his two PO PCAs. There's two because the first one is Curtisi, the second one is Willikeri. But what you'll notice is that his PCAs actually show quite a bit of geographic structure. And they also have much higher degrees of variability being explained by these PCAs. So the picture in Malaria seems to be quite different from what it is in ovale. In addition, we also wanted to look at complexity of infection to see how many parasites are circulating in any individual isolate. So we did this using the real McCoil, and I'm showing results classified by country. So most of our isolates of PM are monoclonal, but we do see some that have 2 or 3 clones circulating in a given isolate. And you will notice there's one isolate in Tanzania that says it has 9 clones. That is definitely not true. And we're going to have to do some post-processing to figure out what exactly happened with that sample. But we don't believe that there were actually 9 clones circulating in that isolate. And I, again, want to put this in context with our other species that we have examples of. So this, again, is Kelly's figure. So he did this for his 2 ovale species, as well as falciparum data that is from similar regions where he collected his ovale samples. And you'll see the ovale mostly monoclonal with just a couple of polyclonal samples. So possibly we have a little bit of a higher rate of polyclonal infection in PM than PO. And then he has falciparum on the right there, where you'll see that there's quite a bit more polyclonal infection than we see in either PM or PO. Although I will add the caveat that I have not yet done thorough comparisons of my PM samples to PF samples from the same regions. So in conclusion, from this part of the work, our hybrid capture approach worked well to facilitate whole genome sequencing of these 50 Tanzanian isolates, as well as our other 16 isolates that came from elsewhere. Actually, it's more than 16, 26. And we found that our Tanzanian PM isolates were closely related across the country. And even further than that, we saw no clear geographic structure even between countries that are on totally opposite sides of the continent. And we saw mostly monoclonal infections, although some polyclonal as well. In the future, I'm going to also be doing this on the samples that we collected in 2022 and 2023. There will be, before this work is published, a more thorough analysis of the isolates from Cameroon, DRC, and Nigeria. And we'll also be doing a better job of characterizing how parasitemia and the presence of other species in those infections relate to complexity of infection. We'll be doing some selection scans on these isolates. And then finally, we'll be doing some comparisons to PF, PO, and PV, looking at nucleotide diversity and orthologs between those species. So one final thing that I'll talk about just very briefly is that we're now working on sequencing of PO valle from these same studies using selective whole genome amplification, or SWGA. So we have 148 kernicite isolates that we'll be processing using a custom protocol developed by another postdoc in the lab, Wenxiao He, and then 92 Walliceri isolates that we'll be processing using a published SWGA protocol for Walliceri. This will include samples from MSMT, as well as from the transmit study in Bagamoyo, from Cameroon, DRC, and Nigeria. And I have no data to present yet, but hopefully soon there will be something to say about these. So finally, I want to make sure that I acknowledge everyone who made this work possible. So first, our funders, the Bill and Melinda Gates Foundation, the NIH, PMI, and the CDC. I want to acknowledge everyone at UNC who was involved in this work in various ways, particularly my advisor, John Giuliano, and then also our collaborators at Brown, at NIMRI, particularly Deus Ishingoma for getting this whole thing going, at the NMCP in Tanzania, at the CDC in Atlanta, in Cameroon, in the Gambia, at MUHAS in Tanzania, and then also in the DRC. Don't have time to read all these names, but there are many, many of them that I'm very grateful to. So with that, I'm done talking, and I'm happy to take questions.
Okay, sure. Thank you so much, Dr. Zach. That was brilliant. A nicely paced presentation as well. Thank you so much. It's time for questions. So please do type your questions in the Q&A box, and I'll read them out for Dr. Zach to answer. So I'll start with mine. Sorry, I'm going to take... so I think I heard you say when Plasmodium falciparum gets eliminated, the other Plasmodium's prevalence increase, like the PM, PO, and PV. Why is that? Because I'd assume that the control measures against PF would work against PO, PM, and PV. Can you please explain that?
Yeah. So there's a couple of reasons why control measures for PF may not be fully effective for other species. So with PO and PV in particular, we worry about relapse. And so if you're just focusing on vector-borne transmission, and people who have, active infections, you may miss those people who have hypnozoites. And so what can happen is that even if you've gotten rid of the falciparum, there may be new infections of PO and PV that mosquitoes can acquire from people with relapse. So that would be one way in which PF control measures would not be necessarily effective against other species. And then in addition to that, we really don't have data about this. And we actually have a project in Cameroon trying to answer this question. We don't know what the vectors are in Africa of the other species. So we know what the major vectors of falciparum are. We know that that's Anopheles gambiae, Anopheles funestus, Anopheles stevensi as the introduced vector. But we really don't have good data about what transmits ovale, malaria, or vivax in Africa. And so potentially those are other anophelines, and those anophelines may have different behaviors, different habitats that would make them not well targeted by what we're using to target the PF vectors.
Thank you so much for that answer. So there's a question from Tanto Situmorang. Hi, do you think that there's a chance that PM and PO are not detected in some people because of their low parasitemia in comparison to PF?
Yes, so I think that is quite possible and even likely. So particularly when we're using RDTs as our primary diagnostic, I think we're quite likely to miss those infections. And so what this study really enabled us to do is by looking at qPCR data, we're able to find those cases even in people who don't have patent malaria. And so that's really the advantage of doing this, is that we know that it's there because those really low parasitemia cases like you're mentioning probably would not be captured by an RDT and would not even be seen by using microscopy. So really, we do need these highly technical methods, at least for now, to be able to detect them in those low parasitemia cases.
Okay, thank you so much. Now it's a comment. I don't know if you have a response to this comment. It's from Loic Pradel Kojomfoko, who says, "Thank you, Dr. Zach, for your presentation. Note that the principal components 1 and 2 of your PCA just explain less than 10% of the variance. So be more cautious when drawing conclusions regarding this section of your manuscript in preparation." That's from Dr. Kojom Foko from Cameroon. I don't know if you have any response for that.
Yeah, no, I really appreciate that comment. Yeah, I agree that PCA really does not explain very much. So I think we're going to have to do some more detailed analysis to try to figure out what we can say about the population structure of PM. I will say the previous data that I'm aware of that used other methods has also found that there's very little, um, population structure in Africa in PM.
Thank you. And there are two comments from Dr. Vicky Simpson, as well as Dr. Jacob, saying excellent presentation.
Thank you.
And Dr. Richard Pearson is asking, really interesting that there is some structure in PO than in PM. Do you have any hypotheses about why this might be?
Yeah, I agree. It's really interesting. And I have to say, no, I haven't really delved into that as to why that might be. Um, I don't really know. Just that, the previous data has, like I said in the last comment, has shown that PM maybe just doesn't have a lot of structure. It may just not be as diverse as PO. But we will be digging into that much more, and hopefully I'll have a more satisfactory answer later.
Okay. I think there's a repeat question from Dr. Kojom Foko. Did you do G6PD and or Duffy group analysis of patients with non-PF infections?
So in this study, no, we have not done that. I believe going forward, they are starting to do that work in Tanzania. Because yes, that's important to know in terms of radical cure for PO or PV. We would need to know if we can give primaquine or if we need to give tifanuquine. So not for this data, but I believe they're doing it going forward.
Okay, again from Dr. Richard Pearson. Also, I see you used bait capture for PM, but are using SWGA for PO. Why the different approaches in the different species?
Yeah, so we used the bait capture for PM because there was just really nothing available to do genotyping in that species on a genomic scale prior to that. And so John Giuliano, my advisor, had money from grants that we were able to spend to get a custom kit designed. So in the earlier PO work that I presented from Kelly, he actually did use baits. And what we did here that I didn't talk about is we've compared our results from our SWGAs to performance from that hybrid capture method. And I think that the major reason we're doing SWGA now instead of the baits is just because it's cheaper and it also seems to show similarly good performance.
Okay, thank you so much. Dr. Jacob Amalgo is asking, do you think it is likely that the other Plasmodium species may take the PF niche if interventions are successful in Africa in the near future, for example, vaccines, and become a big problem for public health?
Um, I definitely think it's possible, for the reasons I mentioned above, with the vectors and the relapses. And we do have some data from Tanzania that Dr. Jess Lin published that did seem to show that ovale circulates in different times of year than falciparum. So it's definitely possible that we may see, increases in PO in particular in Tanzania if PF were to be eliminated. And the data I alluded to earlier showing that PV takes over is from the Solomon Islands, where they have seen cases of that happening. So I definitely think it's possible.
A question from Dr. Vicky Simpson. What are your thoughts on how non-PF species are going to affect timelines for full eradication of malaria?
Yeah, I think they're going to make it difficult, particularly because PF doesn't have these dormant stages in the same way, so it's much easier to detect PF. Although of course there's still sub-patent infection, but I think there is a real problem with PO, PV, and PM in terms of being able to detect it in these people who have either relapses or persistent infections. And in addition to that, they can trans be transmitted to mosquitoes much earlier in the infection, at least for PO and PV. Yeah, I mean, I think people are not aware of them, and they can be transmitted sort of in the off season in a way that we don't see with PF. So I think it's going to definitely be a big challenge.
There's a question and comment from Thomas Katairo. Thank you so much for the presentation. What is your opinion about the low prevalences of PV? This is the same trend we are seeing in Uganda. We have seen only 2 samples in a pool of 2023 samples. Someone said it's because PD sometimes hides in the bone marrows, so escapes being detected. But I wanted to get your view on this. That's from Thomas.
Okay, yeah, that's an interesting idea. I don't know all that much about the pathology of vivax. So I would defer to someone who knows more than me to address the bone marrow question. But it does, I mean, to me, it seems likely that there just is not that much vivax in these populations in East Africa. I mean, we see, as I'm sure you know, quite a bit of vivax in the Horn of Africa, and we've found some vivax in DRC and in Cameroon. But it seems like there's just doesn't seem to be a lot of it in Tanzania and Uganda. So I think that's probably the bigger story is there just isn't much there. But the bone marrow thing is interesting. I don't know much about that.
Okay. We have about 4 minutes left and we still have a ton of questions to go through. So I'm going to cut at some point. So I'm just going to be quick. There's a comment from Innocent Ali saying, very nice presentation, Zach, with an exclamation mark. And Mutai Sila says, hello, could we conclude that persistent parasitemia to PM could be the only reason for high percentage in others compared to school students and young children? And how many of these non-PF cases are detected in routine testing? That's from Mutai.
Okay, so for the first question, that's a fair point. There may be other reasons why we see more PM in adults. We don't really have a good idea of what those would be. Persistent infection is sort of the most well-known factor with PM, but PM generally is not a well-known parasite. We know very little about it. So there certainly could be other factors at play, and I don't know what they would be, but it's interesting to think about what they might be. For the second question, how many of these non-PF cases are detected in routine testing? I would say almost none. So generally RDTs are based on HRP2 and PF LDH. So mostly there is some cross-reactivity, and a lot of these cases are co-infected with PF. So if someone has a co-infection, it's very likely to be detected, but we wouldn't know that they had, say, PO or PV, meaning that they would not be tested for a relapse infection. They wouldn't get radical cure or anything like that. So mostly, I think we don't know about these infections unless we're doing this type of analysis.
Okay. Looking at the time, I'm going to read the last question. I apologize for those of you that still have questions. I'm sure you can get in touch with Dr. Zach to engage and get answers to your questions. The last question that I'm going to read is from Gideon Johannes, who's asking why the presence of malaria parasite species differs from one stage of a person to another stage, adult versus child.
Yeah, that's a very interesting question. I don't have necessarily a very satisfying answer to that. I think some of it probably relates to immunity. So adults will just have had more opportunities to be infected and will have developed more immunity to given species. But I would say also we don't know all that much about the biology of our non-falciparum species in particular to sort of talk about what might be different in adults versus children other than that immunity question. So if anyone knows more than me, I'd be interested to hear it from you.
Okay. Thank you so much, Dr. Zach. I'm going to put my colleague on the spot if she has anything to say. Christina, do you have anything to say before we close?
Thanks, Babalo. Thank you very much, Zach. It was a wonderful presentation and it inspired a lot of questions from the public. I had a couple of questions, but it was already asked by Richard and Jacob, which was really good on SWG and the future of non-Plasmodium species. But thank you so much for your presentation. I think the non-PF species, we'll see the story panning out as we move along. And I think it's going to prove to be quite a tricky thing to tackle in terms of malaria elimination. But we really, really appreciate your time and your wonderful presentation. Thank you.
Thank you so much.
Thanks, Christina. Dr. Zach, any last words from your end?
No, I just really appreciated having the opportunity. It was great to talk to all of you this morning. Lots of great questions.
Thank you so much. There's more questions in the chat, and Q&A, but I'm looking at the time, so apologies again for that. So do get in touch with Dr. Zach on the side. Thank you so much, Dr. Zach, for your time and brilliant presentation. And thank you to so much, all of you, for attending today's Journal Club. There'll be another one in February. We're going to advertise it in due course. For now, the recording for this presentation is going to go up on YouTube. And you can also catch the other 4 presentations that were previously presented in this series as well. Thank you so much, everyone. Have a good day or good evening, depending on where you are in the world. Thank you so much. Goodbye.